So we're moving on now to look at oncogenes. Oncogenes were presented in the previous chapter as genes carried by viruses, which by themselves somehow can initiate the formation of tumors and neoplasm, resulting in some cases in the development of carcinomas. So if these genes were all picked up from the genomes of the host cells, then the suggestion is that there may be other genes within the genome of a cell that may be responsible for similar types of outcomes. So the investigations began in the 1970s and 80s, and they were looking for similar genes which could be then carried by viruses to encode cancer. These are the learning outcomes for this particular chapter. So the historical perspective combined with the current theories of carcinogenesis are important. So understanding the history and then how that history impacted current day understanding is the most fundamental thing that you wish to take away from chapter 4. In addition, understand how to find non-viral transforming genes and how to clone them. So the techniques for finding and searching the genomes for these genes is instrumental too. So this is a great opportunity for us to relate to some of the more common molecular biology techniques that have been employed in the past and are currently employed. Explaining things is a great way to relate to things. So explain how oncogene-activating mutations can be found on different chromosomes and how, in particular, the Ras, or the Ras, pronounce it however you like, was discovered, right? And then contrasting means comparing. So contrast the different mechanisms of oncogenic dysregulation. I mean, this means not proper regulation, and be able to give examples of dysregulation and their outcomes. So this is very important. Can you predict what would happen if a particular oncogene misbehaved? Next, describe the role of translocations in leukemia and as a means of targeting chemotherapy or other types of solutions for people that have these types of mutations. So let's get started. So it's very important that you understand that from the historical perspective, it was highly suggested by the research on viruses that we should focus on the presence of viruses inside human cells in order to have a better understanding of how cancers form and therefore probably how cancers could be treated. So when viruses transform cells, we call that viral carcinogenesis, right? But there's another train of thought that materialized at the time that maybe because we can't find viruses in human cells and their cancers, maybe there's a non-viral explanation of how cancers materialize. And so the two camps started looking for evidence to support their particular positions. And that led to the retroviral theory of carcinogenesis, which says that endogenous, that means residing retroviruses that live within the chromosomes of our genomes, they are responsible for generating cancers whenever they are activated, right? So endogenous proviruses is simply a state in which the virus is integrated into the chromosomes of the host cell. So they said that these chromosomes were passed from generation to generation, and then whenever triggered by some type of event, maybe a stress for the cell or the presence of a certain chemical or physical mutagen, these proviruses were awoken. And when they woke up, they were able to then do their nasty deeds and generate tumors and cancer in that particular population of cells. So let's talk about this. Endogenous proviruses. We do know that this theory has a lot of support because up to 45% of your genome your genome is made up of transposable elements, which in itself is fascinating. But we don't have the time to go into that. The important thing is that 8% of the genome is derived from sequences which look like retroviral precursors. So it's possible that in the past, the distant past of human evolution, these retroviral infections were quite prevalent. But since then, the evidence suggests that they are now beginning to subside in frequency. We're not sure why, but in the human lineage, there's been a dampening down of the ability of these retroviruses to become activated, unlike in other primate species where they continue to cause rearrangements in the genomes. Going beyond primates, many mammals also have their retroviral activity uncurtailed, right? That means in mice, we still see evidence of modern day retroviral activity. The book wants to focus on a particular class of these endogenous retroviruses, and those are the ones that are present at a higher level in the germline. So in the past, there's evidence that these particular retroviral subfamilies were active, and we call those the human endogenous retroviruses. And we're going to look at the information on that. Experiments performed on mice such as these have been instrumental in directing our attention to the process by which retroviruses can induce tumorigenesis. So on the left-hand side, we have a normal mouse, and then we infect the cells of that mouse with a known retrovirus, and then the retrovirus somehow makes its way into the gametes, where it becomes a provirus. So it can be detected using various techniques that we'll look at in the next few slides. And as those gametes are then passed on to the next generation, all the cells of the F1 will therefore carry the provirus. And after many generations in the future sometime, there may be an activation event that causes the endogenous provirus to wake up, and then in that particular cell and its lineage, it will then induce the formation of cancers. And those cancers then can be manifest and detected by scientists. So here we see an example of this happening in real life. So it does happen, but the question is, does it happen with humans? And that's what we want to answer. So moving on to the next slide, you can see the techniques that we want you to learn, such as gel electrophoresis, southern blotting are very important, and we'll talk about them on the next couple of slides. But let's just stay with this experiment here. Let's go for the right-hand side first. So on the right-hand side, we have a number of cells from humans, and we probe those cells looking for the human endogenous retroviruses. And we find that we can detect their presence using various probes that are designed to detect the sequence of DNA that is present in these proviruses. You can see here between different cell lines, there's not much variation. So this tells you that these things are quite unremarkable in that they're not moving around very much. But compare this situation with that of mice. Now, in lab strains of mice, we can see different strains have over the years been bred to possess different types and numbers of these same retroviruses. And in outbred strains of mice, these are the ones that we find in the wild, we can see the patterns vary, in some cases very dramatically, between different strains, suggesting that at least in these mice, the viruses are moving about the genome at a much, much, much higher frequency than we see in humans. So experiments like this, you must be able to interpret simply by looking at the data. And one suggestion is always start with the title, and then from the title, look at the axis before you interpret the data. So fully understand what's going on with the terminology. And if you don't understand that, don't look at the data until you're confident that you can predict and explain to somebody else what this legend on each side means. That's a great way then to be able to interpret the data. So these human endogenous retroviruses are very useful for studying evolution, not just in humans, but other related species. How manipulated the genomes of these particular species have become. Going on below are some of the biological consequences of acquiring these endogenous retroviruses. You can see they're long terminal repeats. You saw that on the previous slide. These are long terminal repeats. These long terminal repeats, they can act as transcription regulators. So if they happen to sit down on the genome of that particular species and happen to be close by to another gene, their transcription products can then influence the rate of transcription of certain genes. And they can also be involved, as we believe, in resistance to viral infections. So having these HERVs inside the human genome may actually dampen down the retroviral activity. So the last thing here is that these are postulated to be cofactors in several diseases, not the primary cause of the disease, but these are cofactors. So you can see multiple sclerosis and schizophrenia on this list, as well as cancer. So the alternative theory in the 1980s was the theory of carcinogenesis not caused by viruses. So these are the non-viral carcinogenesis theories. And they said that it's not a virus that's responsible for initiating cancers in tissues, but other agents. And in some cases, they implicated growth factor genes, so genes that normally regulate the behavior of cells, as we'll see in the next few moments, to only divide when necessary. Maybe those genes have been hijacked by some system or activated through mutation that results in extra chromosomal, extracellular, extra proliferation of cells, right? That's what I meant to say. So the cell cycle is activated. And also mutated growth factor genes are then called oncogenes. So these are the normal genes are called proto-oncogenes. Proto means normal in Latin, and oncogene simply means a cancer-causing gene. So the experimentation presented in this chapter is very important. So this is one of the experiments, and here we're looking to find evidence for these non-viral transforming genes. And what they did was a sequence of experiments where they transfected, transformed certain skin cells from the mice using a mutation, and then they were able to mix the mutated DNA with these cells using a transfection protocol that uses a calcium phosphate or a shock of electricity in combination with calcium phosphate and potassium phosphate to induce the surface of the cells, these cultured cells, to uptake this exogenous DNA. So some of these cells will have taken up the DNA, not all of them, but some. And then what you do is you apply growth factor, and that growth factor may result in the formation of focus. A focus is simply a clump of cells that can be seen on one of these growth plates as a multi-layered entity. You take the focus and you inject it into a recipient mouse, and then you wait, and then you can see that the mouse will develop a tumor. So these suggested that you could have mutant DNA cause cancer directly rather than going through a retrovirus. These experiments came up with some very powerful conclusions. As we can see here, we have some transformed mouse cells based on the previous experimental procedure. And in this case, we have bladder cells. And these bladder cells, they form in vitro on a Petri dish, they form these foci, or focuses to be singular. And once you examine the cells within the focus, you discover a couple of things. The first thing is that the cells are anchorage independent. And the second thing is those same cells, when transferred into a recipient mouse, will cause the formation of tumors. So the question that was asked is, how many genes are involved in transforming these cells? Was it just the number of genes on the transfecting DNA? Or did that trigger other genes to cascade and cause these phenotypic changes in the cells? So that's what we want to discover. And the answers are fascinating. Now, I promised you earlier that you need to know some techniques that are instrumental in historical evidence as well as modern day research. So the southern blot is when DNA is separated according to size, normally along a gel, electrophoresis. And then to make that permanent so it can be used again and again by the scientists, Southern came up with a technique where the DNA can be removed from the fragile gel and affixed to a more permanent matrix. In this case, something like nitrocellulose or nylon membranes. And the DNA simply siphoned up, straight up at 90 degrees from the direction in which it was moving. And overnight, the action of the wicking paper absorbs the DNA onto the nylon. It can't pass through the nylon because the nylon pores are too small, but because the DNA is single-stranded, it can be easily permanently fixed using its backbone by putting it in a microwave for a few seconds, and that's it. And then the filter can be used again and again for many research experiments. So if you do it with DNA, that's called Southern. If you run RNA on the gel rather than DNA, then it's called a Northern blot, simply because it happened to be the opposite of Southern's name on the compass is Northern. There's also a Western blotting. That's for proteins, and we'll come across that in future chapters. There's no such thing as Eastern blot at this time. So results from these experiments where probes are used to identify along these gel lanes various molecules by hybridization. You can see here, the scale on the side is in kilodaltons. So this is the size of the DNA fragments. And you can see we have different strains of mice DNA being run along this particular Southern gel. And here we're looking for telltale signs to show us if DNA has been picked up from the exogenous fragments and how that then changes the size of the DNA fragments themselves. So you can have a DNA fragment the size of my finger, and if it picks up a piece of exogenous DNA, then that size of that finger will increase, and that will be reflected in a shift in the size of these bands. There are a couple of controls here too, so make sure that you understand that. So the right-hand side DNA is from non-transfected cells, whereas the others are from NIH3T3 transformed cells, okay? So make sure you can interpret this in terms of each and every row. That's very important. So the evidence currently is this, that retroviruses can, in some cases, amplify, depending on the species, their genes, right? So retroviruses can carry oncogenic material, but only in some species are they active. But in other species, like humans, they seem not to be active. As the book says, there's only 2 cases of human cancers, cervical cancer and liver cancer, where there's a regular indication that it's caused by a viral agent. Most other parts of the body suggest otherwise. So it's more likely that human cancer is not caused by viruses, unlike in mice and birds, which it seems to be. Okay. Regardless, here's a list of some of the most common bird and mammal retroviruses. And we've come across some of these already in the previous chapter. You don't need to memorize any of them. The point is that the species that they cover have been mentioned in chapter 3 already. And we'll talk about what type of oncoproteins are produced in the next few chapters. Okay, so chapter 5 will go heavily into looking at the receptors, and then chapter 6 will go and look at some of the signal molecules, the growth factors themselves. So we'll get immersed in this knowledge eventually. In this particular case, the experimental procedure is to localize human oncogenes. What you do is you rely on allele-sequences. Allele-sequences are repetitive DNA that are found in 1 million copies in the human genome. So 1 million copies on average means that they are found every 5,000 bases, which is great. So you can use them as a probe to localize any human DNA that's been moved into mouse cell lines. So here we have a potential oncogene whose location we don't know, but there are allele sequences in the vicinity that will help us then track this gene down. You transfect fragments of the human DNA into mice and wait until they form tumors. Once the mouse has formed a tumor, then you breed those mice again and again until you have secondary transfected mice. So the logic is that every time that you breed the mice, the recombination mechanism reduces the size of these fragments. So mice that get the normal fragment without an oncogene will not become transformed, whereas mice that get the oncogene, they should become transformed. Okay, and then all you do is make a probe to the red allele sequence, which is going to be different to the mouse's repetitive DNA, and therefore you can then use that to localize this fragment by cutting the mouse DNA into small pieces and then probing with an allele probe. So this way you found your needle, and therefore many oncogenes are localized, found in this particular manner. So that in itself is fantastic. So the logic behind the previous experimental procedure is based on this Chinese division, where you have a normal oncogene, proto-oncogene, and then you have the oncogene that's cloned, okay? And both of them can be transfected together into mice, and due to random recombination, there will be some mixing and matching of the fragments. And what you're trying to see is if a particular fragment piece leads to the formation of tumors in those mice. And if that fragment doesn't, then the entire sequence, the entire region can be then ignored as a cause of carrying the oncogene. Then your focus lies on the left-hand side in this case. So by doing this again and again, you discover there's a fragment that is always moving with the formation of tumors. And therefore that's the part of the clone fragment that you need to focus your attention on. So again, very instrumental in allowing us to discover tiny fragments of nucleic acid in a vast sea of genomes. Using these methods, we discovered the Ras proto-oncogene, right? And Ras is going to be very important. It's a gateway gene that produces a protein called Ras, and that protein has many active sites in that it senses a lot of things within the cell. So it's like a supervisor monitoring lots of different conveyor belts and making decisions as to whether the cell should move into the cell division cycle or arrest itself until it's fixed any issues. So that's a good analogy. We'll come back to that in the future. Once Ras was discovered, the human Ras gene, the question then became, how did the normal gene, the proto-onco Ras gene, differ from the oncogene Ras. And interestingly, all the research suggests that there's a single base substitution, a single SNP, single nucleotide polymorphism, that is present in the 12th codon of one of the reading frames, and that causes a glycine to be substituted into a valine. And that's the only change that has been detected in a number of these oncogenic studies. This in itself is quite amazing. So it seems that the shape of the protein may be altered by this substitution event. And that change in shape is responsible for the initial phases of transforming that cell into a tumor cell. So this reminds us that the basis of cancer appears to be mutation. So here's one type of mutation that has now been tied up to a particular type of cancer oncogene. We can see there's a table here that shows you how important the Ras gene is in various types of cancer. So pancreatic cancer, you can see in 90% of the cases, the point mutation is in the Ras gene of this exact type, right? Whereas in kidneys, less so than other types of cancer. So this tells us that cancer is a heterogeneous set of diseases, some of which have a genetic basis such as this for the etiology. So now that we know something about the difference between normal proto-oncogenes and cancer-causing oncogenes, what are the major mechanisms by which the final product of these genes could be disruptive to a cell? Well, as you can see here, there's three main mechanisms. Amplification, when the number of copies of that gene or its product becomes increased. Then we have insertional mutagenesis, which we learned about in chapter 3, where something comes and sits inside either the promoter or the regulatory part, or within the gene itself, the coding part. Or, as we'll see today in this chapter, translocations are another major mechanism responsible for causing human tumors, where entire segments of chromosome are moved to other locations within the genome. So let's go through this list here. So amplification is very important. Amplification means that you have more product than nature had intended, right? So you could have more genes, and each gene could be producing the same amount of product, but now you have 3 copies of the gene rather than 1. Now you have 3 times the product. That's one way. The other way amplification can work is that the gene is activated at a much higher level than nature had intended. So for one gene, instead of producing 10 messenger RNAs, you're now producing 1,000 messenger RNAs because of some type of regulatory failure. So that can also cause amplification. There's a third way in which normal proteins in the cell may have a certain life expectancy, and because of some mutation, that life expectancy is now increased so that the number of proteins inside the cell begins to increase in sheer numbers, right? For instance, a protein would have a life expectancy of 3 hours. Well, because of a mutation, it now has a life expectancy of 3 days. So as more protein is made, the protein that's supposed to be recycled back into amino acids is not being recycled, and therefore it continues to work within the cytosol, causing an amplification event. So where do we find examples of this? Well, let's look at the left-hand side first. So the N-myc gene, which we came across in the past, its amplification was correlated with a neuroblastoma in children. So these are pediatric chromosomes, and you can see there are whole regions that have been probed for the presence of this gene, and it lights up huge amount of this particular pair of chromosomes. So this is called homogenically staining regions, right? because they stain a single color rather than a single spot being stained. The entire chromosome is stained, or the segment of that chromosome. That means every part of that chromosome must be amplified for it to bind to that probe, which should normally bind to one sequence on each chromosome. So that's a form of amplification. And there's a, as the book says, there's a poor prognosis for children that have this particular cytological outcome, unfortunately, right? How about another type of amplification? Well, we see that the same gene, c-Myc, is a cousin gene. So N-myc and c-Myc are slightly different genes. So c-Myc amplification can also be correlated with poor prognosis in breast cancer, right? And we can stain for that to see how many proteins we have of that gene product. And here we have normal cancer cells, and there we have the same cancer cells now being probed for amplification. And you can see in some of them, we have multiple spots lighting up rather than just two, indicating amplification. What's the correlation? Well, as we said on the previous slide, copy number is associated with survival. So those patients that have low copy number of the N-myc gene, they tend to survive longer than those patients that have greater numbers of the c-Myc gene amplified. So if you have more than 10 copies, your prognosis is pretty poor, and that most of the patients have succumbed to their disease by about 18 months, whereas most patients that have less than 10 copies are still alive after 7 years following diagnosis. So graphs like this, or charts like this, are going to be very important throughout the book in comparing the outcome under different circumstances. Let's talk about a particular oncogene. So the ERBB2, or HER2, also known as NEW, and the reason there's multiple names is because multiple scientists happen to be working unknowingly on the same gene. And then later on they discovered, oops, this is the same gene. So the names have carried over from previous research. It's a historical thing. Regardless, right? This particular oncogene, once it becomes onco, produces a protein. And that protein can be detected by both southern blotting and northern blotting, and by something called immunoprecipitation, where antibodies are used to bind to the protein product of the gene. And you can see down the bottom here that there's a number of different types of outcome. So in the top, we can see that this particular breast cancer binds to more of the probe than does this one. So that indicates that the amplification event is much greater in this tumor than in that tumor there. And these two tumors are in between, okay? With this one being slightly more and this one being slightly less amplified. So that's one scenario where the gene has been duplicated itself. As I said earlier, you can also have the same gene, but present at one copy, but producing lots of messenger RNA. So here it suggests that the regulatory region of the gene, the promoter or the enhancer, has been compromised. And now, even though you have one copy of the gene, it's active at a much higher level, therefore producing lots of RNA, okay? So that's another telltale sign that something's wrong with the biology in that particular cell for this particular HER2 gene. And then, of course, you can also measure the protein complement of that cell directly by immunoprecipitation. So the same cell line, the same cancer, you can see there's a greater amount of protein produced from these greater amounts of messenger RNA, and that is indicated here. Whereas this, it seems to be the same amount of one copy of the gene with a normal amount of messenger RNA with a normal amount of protein being produced. So maybe something else is wrong in this particular cancer. But the others can be explained by a defect, either at the DNA level or the RNA level, right? And there's also another possibility that, as you'll see, based on what we learned in chapter 3, RNA interference, that could also have another outcome that you need to keep remembering. So looking at the same type of outcome here with HER2, we understand that the survival depends on amplification or no amplification of this particular gene. So here we have more than 5 copies, and here we'll have just a single copy. And you can see the outcome for the patients over 72 months. Even though they approach each other at the end, the survival of the patients is drastically reduced when you have more than 5 copies of HER2. Okay, so this was done over 11 cancers, and this data was generated over 52 different cancers in patients, right? All right, so here's some information that goes with the previous slide that talks about how the receptor at the cell biology level works. So if you want to read that, you can now, but in the future, we'll be covering this anyway in chapter 6. So what are gene expression microarrays? Well, these are simply techniques that are designed to allow us to measure the relative amounts of the product of certain genes. So doing it the old way in the previous 2 slides can be tedious. And now we have stronger technology that allows us to screen lots of samples simultaneously to look for patterns of not just a single gene being overexpressed or underexpressed, but looking at multiple genes within that sample. So these are newer techniques that use DNA chips. And you can see, if you go through this, it explains the logic behind microarray analysis. So what does this mean? Well, here we have 360 tumors examined from different patients. And all of these tumors have been, all of these fragments of the DNA have been aligned by the computer to just focus in on chromosome 17 and its large arm, all right? So the large arm of chromosome 17. Now we're going to be looking in the vicinity of HER2. So HER2 sits right there. And what we're doing in this case is we're looking on either side of HER2, above HER2 on the chromosome and below HER2, trying to find a pattern. The way the system works is this. If the level of the gene is normal, as in the wild type tissue, then you get a green signal. If the level of the gene in the tumor is higher than wild type, then you get a red signal. So you can see here in about 1/5 or so of these tumors from these 360 patients, we can see that there's a neighboring effect. So not only is the HER2 gene expressed at a higher level, but neighboring genes are too. And the explanation for that is maybe the entire fragment of this chromosome has somehow become amplified, therefore leading to more product. So what you're trying to do is correlate this with surrounding genes to see if there are other genes in the vicinity that may also lead to an increase in the formation of these cancers. So you're still looking for that evidence to have a comprehensive understanding of which combination of genes, if they become defective or amplified in any way, can lead to the generation of cancers. So this is how primary research is conducted, where you don't have a good idea where to look. You just look at what you think you know and then look around it to see if there's any association. Right? Association is a very important thing in genetics. So what are we learning? We're learning that a cell is very sophisticated. It uses chemical messaging all the time through multiple pathways to control its biochemistry. So we have the metabolic biochemistry, and then we have the cell cycle biochemistry, and then we also have the cell sensing biochemistry. They're all intermingled, but they are divided into nice compartments like this. So What have we learned? Well, the important thing is that when a particular receptor like this one, the HER2, becomes non-normal, becomes abnormal, it can lead to a number of different pathways within the cell being simultaneously altered. Okay, so one result of this is that there's increased angiogenesis within that region. So angiogenesis is the formation of blood vessels, right? So blood vessels are very important to supply the expanding number of cancer cells with fuel, oxygen, and metabolites. The next thing is, in some other cases, mutations of this particular receptor have led to the greater ability of cells to migrate away from the original location to new locations. So that's metastasis. And another consequence is that the cell cycle is no longer coordinated in a way that prevents the cell from going out of control. In addition to that, the ability of mitochondria to release toxic substances to kill the cell, known as apoptosis, those are blocked, so that the suicide program, the apoptotic pathway, is nullified. So all of these events are key to developing cancer. So can a single mutation result in this profile? Well, that's the goal of this particular course, to find out what the standard way in which cancer develops. Is it a one-step process, or is it a multi-step process? And if there is a multi-step process, how many steps are necessary in order to take a normal tissue and transform it into cancerous tissue? So these are wonderful insights into how the bucket of cancer diseases, lots of them, how they work. It could be that one type of cancer may work one way, and a different type of cancer may work in a slightly different way, or a completely different way. So let's see if we can answer that question by the end of the course. So using antibodies, we can track down and bind to these oncogenic proteins that are produced by these different genes. So you can buy these antibodies, or you can share them with other researchers, and they're becoming available both commercially and through voluntary donations. And they can then be used as a treatment for binding to and neutralizing any oncogenic products that shouldn't be there in that cell. So imagine you're a normal cell, you may have 10 doses of this protein, and everything works normally. Now you're a cancer cell in a patient, and instead of having 10 copies of that protein, you have 100,000. So what these antibodies can do is to bind to the protein and basically stop it from doing its naughty business, therefore neutralizing the effects of that amplified protein. Maybe the tumor cell will then go back to behaving normally. So that's the emphasis of this type of research, right? So these are available from the FDA, and more of these are beginning to appear as an alternative treatment to chemotherapy and radiation. So let's now move on to insertional mutagenesis using viruses, which, like we said earlier, you should be familiar with, right? So this figure came from a figure that was presented in chapter 3. And you can see here, this is the normal genome of a retrovirus. And now this is a normal proto-oncogene in an infected cell. Somehow, in the formation of progeny viruses, some of this will be incorporated into this. And you can see the hybrid molecule down here, and it contains a copy of this, which over time has sustained its own mutations, and then it creates a potent oncogene if this particular genome of this virus happens to get into that particular cell line. Okay, so this is how nature has allowed genomic DNA to be sequestered by viruses in an accidental incorporation. Now, in most cases, the presence of something here would be harmful to the virus, and the virus won't be able to replicate properly. But every now and again, the stars align, as they say, and a perfect fragment of the genome is picked up in the right configuration to still allow this to function as a virus, a retrovirus, and it then gives it an added benefit, right? So think about it. If you're a virus and you pick up a proto-oncogene and you happen to be able to manipulate it through random mutations, how will it benefit you? And the answer is, well, the more the cells proliferate with you resting inside their genome, the more copies of that genome there are for you to grow in your population. So you are basically favoring your particular species of retrovirus. And that's the underlying basis of natural selection. Okay? Although natural selection applies to living things, you can also apply it to the non-living viruses. So do we still find viruses implicated in human cancer? Yes. And do viruses cause human cancer? In some cases, yes. And you can see in this list here, most of the tissue is going to be lymphoid tissue, part of your white immune system, the white blood cells, and your lymphoid tissue. The lymphoid tissue will be the B cells and the T cells. And the other part here is hepatocytes, which are liver cells, right? Those are the main targets of these viruses. And you may be familiar with some of these types of malignancies. Burkitt's lymphoma, that's quite common, and that's caused by the EBV virus, Epstein-Barr virus, right? But you can see you can also have some types of herpes virus as well, including human papilloma virus, right? Which causes cervical epithelial changes. So that one there is a very big problem in schools at this point, high schools. I hate to say it, but because of oral sex in taking place in bathrooms, that seems to be a rage at the moment. So doctors are particularly, pediatricians are particularly worried about that behavior because it is leading to an increase in cervical as well as throat and mouth cancers, because that virus can do its dirty deeds that way. So let's look at translocations. So this is the main part of the end of the chapter. So this is where the focus lies, right? So what they begin the story by looking at is a correlation between a certain type of cancer of the white blood cells called Burkitt's lymphoma and translocations. And incidentally, the story is fascinating. It says in parts of the world where the Anopheles, I think it is mosquito, the one that carries malaria parasite, is endemic, there seems to be a correlation with the Burkitt's lymphoma incidence being very high in those parts of the world. So the story in your chapter is fascinating if you'd like to read it. So the question is, which came first, the chicken or the egg, right? So have a look at that and we'll see if we can decipher that by the end of the course. But the bottom half of the figure is very important. What it's saying is that what we observe in Burkitt's lymphoma in the majority of cases is a translocation, a balanced translocation. What that means is the bottom half, the bottom tip of chromosome 14 switches places with the bottom tip, in this case, of chromosome 8. So this part here breaks off right there where the dotted line is, and it moves and it binds to the exact location where the dotted line there breaks. So there's a balanced exchange of chromosomal parts. So you would think that doesn't make any difference because the genes haven't increased. All that's happened is the location has moved. Well, the question then becomes, where have you moved to? You may live on this street where things are quiet, and you may be moved, your house may be moved to another street where things are rowdy. Now, in that new location, you may be encouraged to join in the rowdiness and then change your activity. Yes. Isn't that a form of amplification? It seems to be, right? And the reciprocal could be the same. So you live on a rowdy street and your house gets moved to a quiet street, and now you're in a quiet environment, and then you're under pressure to quieten down. So maybe that's what's going on here. Or it could be that the breakpoint, this dotted line, is within a gene or its regulatory region. And if it is, that gene now becomes broken. It gets broken up into two parts. So that gene can no longer produce a product, or maybe its regulatory part no longer works. Maybe the place where you go and move to, you start listening to that regulatory region. So you can see how translocations can result in a change in the biology of a cell. On this particular slide, we're going to look at this movement of DNA from one location to another. And in this particular example, the breakpoints are hotspots. That means they're very, very much preferred over other regions of the DNA. So it's not an entirely random process. So when these breaks take place within chromosome 9 and between chromosome 22, there are these hotspots indicated by these dotted lines, okay? Now suppose the breakpoint is there, and then you get reciprocal translocation. This is the product that's generated from one chromosome. This is the product that's generated on the other chromosome. Now you can see whichever one of these two is active, and in this case we're assuming the bottom hybrid gene is active. The front of the polypeptide is going to be based on the BCR gene, and the back end of the polypeptide is going to be encoded by the ABL gene. So this hybrid protein is implicated in certain types of leukemia. That is a very common outcome of translocations. So here's another example where we have different combinations of breakpoints possible, right? We have these domains. These domains are very important in binding tyrosine kinases, or tyrosine kinase activity. And by reshuffling the genes, you end up with a different combination of DNA domains or protein domains in the final protein. So this protein does weird things compared to what the wild type situation would have been, okay? So you still have the DNA binding and the actin binding domains, but now these other domains here have been attached to alternative domains. So this protein that's produced here, the P210, is now going to be a super protein. It can do multiple things that it couldn't do before in its wild type configuration. So the last part of this particular analysis is how does the system work at another level? Well, when you make hybrid genes, you may actually end up producing hybrid proteins. But in the case of this particular gene, HMG-A2, that has been discovered to work in an alternative way. So this particular protein product isn't so much a fusion product. The question is, why is that fusion product made at such a high level? So we're looking at a type of RNA interference. So you can see that over here. So the best thing to understand here is that messenger RNA can be intercepted by microRNA. So microRNA has to be produced properly, and if it's produced properly, it can then bind to the RISC complex and then attenuate messenger RNA so that the amount of protein is reduced, right? So this is the policeman, and the policeman binds to the messenger RNA, and then it degrades the messenger RNA, therefore reducing the amount that approaches a ribosome and then can be encoded. This is something that we would have learned about in chapter 2. Because of this reciprocal exchange, now the gene is missing the binding site for the RISC protein. So the RISC protein no longer can find that because it's missing. And therefore, the hybrid protein gets away with being degraded, the messenger RNA, and therefore it does reach the ribosomes, and it's encoded at a much higher capacity than the cell had intended. So we have an increased activity of that protein using this RNA interference mechanism that is defeated by the hybrid protein, or the hybrid gene to be more precise, right? So the presence of the hybrid gene, this is the normal and this is the hybrid. You can see here everything happens normal, and on this side, everything is abnormal. So if you understand this figure, good. If you don't, please take a few minutes to understand the left-hand side first, and then move your attention to the right-hand side. So what seems to be one of the consequences of these proteins being defective? Well, one of the consequences, as indicated on this slide, is that you could have a misformed receptor protein. Cells are constantly listening to the environment outside, and they are normally paying attention to growth factors and signals that encourage the cell cycle. If those signals are amplified or they are turned on accidentally when they shouldn't be, then the cell knows no different, and it just does what it's supposed to do, which is to enter its cell division cycle. And that's a sign of one of the telltale signs or hallmarks of cancer, is uncontrolled cell division. So how does that work? Well, here's a normal cell, and in this case we have a transmembrane protein. The protein has a extracellular set of domains as well as the cytoplasmic domains, right? And then it passes through the plasma membrane, the transmembrane domain. So under normal conditions, the activity of the protein inside is only turned on once it binds a suitable ligand. In this case, the ligand is a growth factor. What we're discovering is that if, for some reason, the ligand binding domain is missing because of translocations, then in some cases, the protein escapes quality control standards inside the endoplasmic reticulum, and it makes its way to the surface. And if it does in those cases, which are rare, but it can, if it makes its way to the surface, the protein by default may be on, even though it's not binding any ligand. So as far as the cell's concerned, it's getting signals to divide, and therefore this cell will divide uncontrollably. So you can see these mutations that lead to protein changes can also result in cell proliferation. So we're going to spend a lot of time in the next chapter looking at these receptors. So for now, just pay attention to this one. So Gleevec, which is something that Lance Armstrong was given for his testicular cancer, is a drug that binds to proteins and neutralizes their activity. So in his case, he had this CML enzyme present in huge numbers because of his cancer. And what they were able to do was to bind Gleevec into the binding site, the active site, for ATP, therefore preventing ATP from docking and activating the cascade of signals that were driving his cancer. And then by giving Gleevec, you're able to block that activity and therefore tame the cell back down to normal levels. The important thing that we're going to learn is that these pathways, unfortunately, are very complex. They're not simple. We're still trying to elucidate how they work. So it's like a spider's web of interactions. You pull over there and everything over there shakes, right? But it doesn't mean that we stop investigating. And the more we investigate, the more we learn. And the more we learn, the better the treatments are for cancer patients. So you can see benefits from modern research are very, very much on the forefront of cancer treatment. This is from a science paper that was published about 20 years ago. And here we're looking at AML, right? So this is a type of leukemia that is acute myelogenous leukemia. This is prevalent because of these translocated gene products, right? And you can see in some cases, we know what the fusion product is. In other cases, it's a random set of translocations. In others, there's no association between the disease and the cancer, right? And the genetic process. But in many others, we can see that there's some patterns that are correlated with chromosome breakage and rejoining. And that's going to be important because if we can discover what the product of that translocation is and how it impacts the cell biology of those cancer cells, maybe we'll have the ability to block the excess production of that signal. Therefore, that might be a solution for that patient. Okay. So how much of this have we covered? Everything. Are you able to spend some time and relate all these learning outcomes to this particular chapter? Hopefully you can. If not, office hours are available and I welcome you there. Thank you so much.